Welcome, my name is Ben Westhead and today I'll be discussing a research article that investigated the incipient resistance of Helicoverpa punctata to the cry-2AB protein found in Monsanto's Bollgard 2 transgenic cotton. In this article, published in 2010, it states that 3 researchers involved measured the frequency level of resistant alleles to the toxin in the main cotton-growing regions of Australia. This research was valuable in understanding that the pyrimidine of dual toxins is not foolproof and justifies the work for monitoring for resistance within the wild. The researchers' aim was to measure the level of resistance to the toxin cry-2AB in the Helicoverpa punctata populations of cotton-growing regions in Australia. Within this research, there are 2 main methods of measuring this resistance frequency. The first is an F1 screen, where a field moth reared from egg or larvae is mated with a moth-known resistance, which has been raised from a resistant laboratory strain, HP-14, and measures only 2 alleles. The pairs mating results in a bioassay that shows the field moth's allele configuration, i.e. if the moth was fully susceptible, all the offspring produced are heterozygote and are affected by the protein. If the field moth was already heterozygote, 50% of the offspring should be homozygous resistant and 50% heterozygotes, etc. Within the F2 screen, the method is the same, and the 2 unknown field moths are mated together, and the offspring are reared, and then their offspring are tested for resistance to cry-2AB and measures 4 alleles. In the research, the F1 screens were performed on populations found in cotton cropping and non-cotton cropping regions, whereas the F2 screens were performed on collections from the main cotton-growing regions. The results shown in the research article are from the 2004 to 2009 seasons and were from the F2 screens performed on live specimens from cropping regions and measured an increase in resistance frequency from approximately 0.002 to 0.012, a difference of 0.010. These results prove that resistance is inevitable regardless of the introduction of dual biotechnology measures. The data from the F1, which are not as complex as the F2 screens, was used in comparison to the F2 data to provide a distinguishable difference in frequency of resistance between cropping and non-cropping regions. It was found that there was about an 8-fold increase of resistance frequency between the 2 regions. Within the article, the discussion explains that there are now 2 forms of evolutionary field resistance to dual toxin cotton. Firstly, being a significantly greater number of resistant alleles in cropping regions in comparison to those non-cropping regions. And secondly, the gradual increase of resistant alleles found in cropping regions across the 5 seasons measured. The discussion also points out the importance of a 3rd agent to be used in the fight against pest species found in Australian cotton. This 3rd agent needs to be specifically targeted to the 2AB resistant larvae that survive towards the end of the season, and that this agent may benefit from not being found in the cry-2 class proteins. Thank you for listening.